The cell cycle is the process of growth and division of a cell. Before the cell can divide, it has to go through four main phases. The first growth phase, synthesis phase, second growth phase, and the mitotic phase. Let's start with the first growth phase. The first growth phase, also known as G1 phase, the cell increases in size and proceeds with normal metabolic roles. After the G1 phase, the cell goes to the synthesis phase. In this phase, DNA molecules replicate. The next is the second growth phase, also called the G2 phase, where the cell reaches its maximum size. Also, chromosomes duplicate to prepare for the mitotic phase. Lastly, we have the mitotic phase, where the cell goes through four main changes to completely divide. The four phases are prophase, metaphase, anaphase, and telophase.
In the prophase, DNA forms chromosomes, which are condensed chromatin. The nuclear membrane and nucleus are no longer visible, and centrioles migrate to opposite poles of the cell, causing spindle fibers to form. These spindle fibers form a protein structure that helps divide the genetic material in the cell. Once this process is finished, the next phase begins. Now we're in the metaphase. In this phase, the chromosomes align at the equilateral plate, and the spindle fibers attach to the centromeres of the chromosomes. The centromeres are the center of the intersection point of the two sister chromatids.
Next, there is the anaphase. The spindle fiber is shortened, causing the centromeres to split. The separated sister chromatids are pulled behind the centromeres and begin to move to opposite poles of the cell. The last stage is the telophase. The chromosomes reach the poles and the nuclear membrane begins to reform before the chromosomes uncoil. The spindle fibers disintegrate and the cell undergoes cytokinesis, the splitting of the cytoplasm. The nuclear membrane and the nucleus finally form and the cell splits completely, creating two daughter cells.